Hello, my name is Dominic Olver, and today I'll be presenting, on behalf of my colleagues, time-dependent osmotic damage of sea urchin to oocytes. Let's get started. The first thing you need to do to vitrify a cell is to have the right amount of CPA in it. To get that CPA inside, you need to have a loading, and then after thawing, you need to unload it. So during this whole time, you're going to have CPA inside the cell, and for all CPAs, you'll have an accumulation of cytotoxicity. So today we'll be covering how you can mitigate that and the implication of osmotic damage that plays into it. Okay, so the rate that CPA enters the cell is proportional to the concentration gradient on the outside of the cell with respect to the inside of the cell. And so the higher concentration gradient you have on the outside, the faster it's going to be pushed inside the cell. And we can check that out. So we're going to have the MSC, so that's the external soluble solution, which this is our CPA, effectively. And the more we give it, the more goes in. And you can see this plateau is it equilibrating. Now let's say we want to get to our goal CPA. So if we were just underneath it and we equilibrate just to it, it takes a decent amount of time to go and equilibrate with it. Now, while it takes all that time to equilibrate, the amount of cells that die throughout that time increases because of that accumulation of cytotoxicity. But you can avoid this amount of time by increasing the concentration. So instead of it taking about 26 minutes or so, now it only takes about 1 or 2 minutes. And instead of having the survival rate out here, you have it up here. So that's an increase of survival. So you have a better loading protocol, right? However, the issue is that the CPA, the more you put it in, the more it actually adds to the osmotic gradient. The osmotic gradient is what pushes water in or out of the cell. In the case of loading, the water is going to quickly leave the cell in a hypertonic or hyperosmotic condition and then come back in with the CPA. And we'll have an example here. So we're going to load it up a little bit. So the CPA flows in, the water flows out, the cell shrinks, and then it starts to equilibrate. So the CPA is going inside the cell, and with the CPA going inside, then the water can come back in as well. And so we actually have a shrink-swell cycle. And the issue with the more concentration you have, the higher, the deeper this dip goes, right? And so if you have it like this, you're going to go down by quite a lot, very sharp. And if you have it with an incredible amount, then the shrinkage is quite high. And let's say your osmotic tolerance limit was 80. That means that for this loading protocol, everyone died. All of your cells are dead because they all got killed from the shrinking damage. And so despite you avoiding the cytotoxic damage, you still have everything dead from the osmotic damage. All right, so the issue before was that doing a single step, so exposing it instantaneously to a high concentration, made the cell shrink way too much. A solution to it is having a dynamic external concentration. So throughout time, we're going to increase the amount of CPA the cell is exposed to. So while the CPA is going into the cell, we put more CPA on the outside. So what that means is that we start out with relatively low external concentrations of CPA, and then we gradually increase it. And that means that our volume doesn't ever cross our osmotic tolerance limit. And we just keep on increasing, and then at around just after about 4 minutes for this cell type, you get your CPA volume by volume ratio, and you never have the volume of the cell decrease past that osmotic tolerance limit. What else that means is that your survival is not bad. So your cytotoxic survival. So you have hypothetically no osmotic damage, since we stayed within the tolerance limits, and very little cytotoxic damage because almost all of our loading was very, very short. So we don't have that accumulation like we had before. So this is one type of optimization that you can do. But the key that it relies on is the assumption that this osmotic tolerance limit is constant throughout time. And it's always this line. Right? Well, sometimes we don't have a method of continuously changing the external concentration, but what we can do is have stepwise changes. And so we take it for a certain amount of time in one solution, and then we step it up to the next solution. And that brings us down, and we can create a bunch of steps, and each step is calculated such that it never crosses the osmotic tolerance limit. However, this idea of a constant tolerance limit is not necessarily the case. Researchers found that time of exposure to hypertonic conditions resulted in a higher likelihood of losing membrane integrity. Here, the white dots are membrane integrity percentage, while the cells are shrunk. The black dots are membrane integrity percentage of cells after return to isotonic conditions. We can see a clear relationship that the longer the cells were exposed to hypertonic conditions, the more likely they were to be damaged after return to isotonic conditions. This means that osmotic damage is not just dependent on maximal volume achieved, but also the amount of exposure time. So this means that protocols which use constant tolerance limits are not necessarily optimized, since those tolerance limits change throughout time. This idea allows us to make an assertion that if osmotic damage is a function of time and concentration, then survival is dependent on time and concentration. I was invited to Spain to work with Estefania at the University of Vigo. I got to do lots of work in the ECIMAT, especially with sea urchins. This provided an excellent opportunity to test the prediction of time-dependent osmotic damage in both hypo and hypertonic conditions. We worked with mature sea urchin oocytes. We harvested from 3 spawning adults and used about 600 oocytes for each replicate. To test swelling damage, we looked at hypotonic tolerance throughout time. We exposed the oocytes to multiple hypotonic solutions by diluting seawater, which is about 1 osmolality, with deionized water to provide the desired osmolality. For each solution, 3 replicates would be exposed for a set amount of time, such as 2, 6, 15, up to 90 minutes. After exposure, cells would be placed back into isotonic seawater and allowed to equilibrate. We would then fertilize the oocytes from a single male. Control replicates were kept in seawater throughout the experiment and fertilized at the same time as the other oocytes. We used the same experimental design to test shrinking damage throughout time. However, instead of diluting seawater, we added NaCl or sucrose to achieve the desired osmolality. Again, after the set amount of exposure time, the cells would be returned to isotonic seawater and later fertilized along with the control. After fertilization, the cells would be left for 48 hours at room temperature in seawater. After, at least 100 individuals from each replicate would be counted. The survival ratio was obtained by the amount of cells which reached the 4-arm Plutia stage divided by the total number of individuals counted, and then normalized with respect to the control. We used a model based on accumulation of volumetric deviance throughout time to predict osmotic damage. I will not go into the derivation of this model, but I will try to provide an intuitive understanding of how this model works. First, J-vol is the amount of accumulated osmotic damage. J-vol is proportional to the integration of the absolute volume difference between isotonic volume, V-iso, against the current volume, V, which changes as a function of external concentration, Me, and time, T. Beta and lambda are constants that we fit. To get the survival ratio, we simply raise the negative of J-vol to E. To predict what volume is throughout time, we use the Boyle-van Hoff model to inform the two-parameter model and fit water permeability along with ethylene glycol permeability. Although not perfect, this provided a good estimate of how fast water flows across the membrane of the oocyte. Since we know what the volume is throughout time, assuming the external solution is impermeable to the membrane, we can now plot the difference between isotonic volume and current volume for both shrink and swelling of cells. Assuming beta is 1, the shaded area is precisely the integral of the difference between volume and isotonic volume. This area is then shrunk or enlarged by lambda. This idea of adding up the difference in area is precisely what is captured by the integration granted the area is modified by the beta and lambda constants. Now that we have a handle on the model, let's see how it fits the data. Here we have the survival ratio normalized to the control. We only fit 2 parameters for the entire swelling damage model and achieved an adjusted R-squared value of 0.97. This means each one of the colored lines all use the same lambda and beta numbers, and our model does a pretty good job at explaining the data. As for shrinking, the hypertonic NaCl solutions showed a different story in the fitted constants. Despite this difference, the R-squared value was still pretty good, indicating our model is successful at explaining shrinking damage throughout time. Hypertonic sucrose solutions were more deadly than NaCl solutions throughout time. Higher levels of sucrose also made the experiment difficult due to higher viscosities and tendency for solution to remain. This increased error in the data, and the model only achieved an adjusted R-squared value of 0.79, mostly due to estimates of solution containing high levels of sucrose. Despite difficulties in design, it was very enlightening to see how different solutions can result in apparent change in time-dependent osmotic damage. Taking everything into account, it is evident that swelling damage works differently than shrinking damage, as shown by drastic differences in fitted constants. Note that beta is a power constant, meaning that relatively small changes to beta can result in very different behaviors. Furthermore, the difference in fits of hypertonic solutions indicate osmotic damage is also solution dependent. Perhaps this is due to differences in membrane interactions with NaCl, which is naturally abundant in seawater, and sucrose, which is not naturally abundant in seawater. Further work is necessary to determine the cellular mechanism of this phenomenon. Currently, our model works well to predict time-dependent osmotic damage. Now, what are the take-homes? The first is that we cannot assume osmotic tolerance is constant throughout time. This means optimized protocols which don't take into account time-dependent osmotic damage are not necessarily the optimal protocol. Instead, they may be over or underestimating osmotic damages, depending on the time frame of the protocol. The second take-home is that our new model provides a new way to develop optimized protocols. For instance, if we simply minimize the osmotic damage for a 3-step loading and unloading protocol, we can prepare an oocyte for vitrification with only 2% of the population dying from the osmotic damages. Finally, our model provides needed insight into approaching loading and unloading protocols, not just for cells, but tissues and organs as well. By combining our new model with existing tissue models, it may be possible to predict optimal protocols which include time-dependent osmotic damage. I would like to thank all my colleagues as well as Sarah Campos. I would also like to thank all the institutes which aided in this work. Thank you.